Imagine that you are reduced to a small size, at a much smaller size than a cell, approximately 0.1 nanometers, maybe even smaller. All cells have in common that they consist of a nucleus, endoplasmic reticulum, mitochondria, microtubules, lysosome, Golgi apparatus, centrosomes, ribosome, cell membrane, cytoplasm. Your journey begins through one of the 200 types of cells in the human body, where you will get to know its parts, functions, and some amazing things. In front of you is the first obstacle, the mosaic structure of the membrane, and the numerous proteins that dance there. There are several types, and you assume that each one has an important job to do. The term osmosis originated in 1827, and its importance to physiological phenomena realized. But it was not until 1877 when the botanist Wilhelm Pfeffer proposed the membrane theory of cell physiology. In 1890, a revision to the cell theory stated that cell membranes existed, but were merely secondary structures. It was not until later studies with osmosis and permeability that cell membranes gained more recognition. In 1895, Ernest Overton proposed that cell membranes were made of lipids. So now let's take a closer look to see what important roles the membrane and its parts play in the physiology of the cell.
A glycoprotein, isotype of conjugated protein, with shorter branched carbohydrate chains known as oligosaccharides. They are part of various biological structures, like the outer surface proteins of cell membranes, plasma proteins, mucus gland proteins, hormones, and enzymes. Glycoproteins also function extensively in the human innate and adaptive immune system. In fact, almost all immune molecules are glycoproteins. For example, glycoproteins form the T-cell receptor complex, the antibodies produced by B cells, and the major histocompatibility complex. Cytokines secreted by immune cells that control inflammation are also glycoproteins. Proteins range in molecular weight from 1,000 to more than 1 million Daltons, DA. But the folded size of a globular protein is not necessarily correlated to its molecular weight. Proteins composed of about 250 amino acids or less often have a simple, compact globular shape. Larger globular proteins are usually made up of two or more recognizable and distinct structures, termed domains or modules. These are compact, folded protein structures that are usually stable by themselves in aqueous solution. Typical domain structures consist of hydrophobic cores with hydrophilic surfaces. Individual domains often possess unique functional behaviors and often perform unique functions within the larger protein in which they are found. Accounting for up to 40% of the membrane, cholesterol is essential for the structure, elasticity, and the various functions of cell membranes. Cell membranes act as barriers that separate the interior of the cell from its surroundings and regulate the selective transport of material into and out of the cell. This makes the membrane thicker and less penetrable. Cholesterol displays a very important function as a component of cellular membranes, especially the cell plasma membrane, where it is found in higher concentrations. Its positioning into the lipid bilayer and interaction with other lipids have a significant role in membrane fluidity, together with other lipid components, such as the amount of sphingomyelin or the degree of saturation of the phospholipid acyl chains. Integral membrane proteins constitute nearly 30% of most genomes and play critical roles in cellular signaling and transport processes. Integral membrane proteins contain large hydrophobic domains embedded in cell membranes, which are essential to supporting proteins' correct fold and function. Integral membrane proteins, IMPs, are essential for life as they transport matter and signals across cell membranes and generate electrochemical gradients. They play key roles in signal transduction, nutrient uptake, and energy generation, and are key targets for drug development. structural and functional characterization of integral membrane proteins is still very challenging due to their low abundance and difficulties in purifying functional IMPs from their native membrane environment. Many IMPs require specifically bound lipids or a lipid environment for full stability and activity. Channel proteins allow charged or large molecules through the membrane. For example, channel proteins allow ions like sodium, potassium, and calcium, as well as larger molecules like glucose, to pass through the membrane. Humans have approximately 400 genes encoding channel proteins. The historical channel nomenclature, based variously on the ion transported, mode of regulation, physiological role, or drug sensitivity, is often ambiguous. Channels are integral membrane proteins, usually with 2 or more A helices crossing the lipid bilayer. A special domain of the ion channel protein that senses the voltage across the cell membrane. Movement of the voltage sensor causes opening and closing of channel gates. Alpha helical integral membrane proteins, IMPs, play essential roles in biochemical and electrical signal transduction, molecular transport, and energy propagation among other critical cellular processes across the membrane. It was estimated that about 1/4 of the human genome encodes A helical IMPs. The alpha helix is the most common structural arrangement in the secondary structure of proteins. It is also the most extreme type of local structure, and it is the local structure that is most easily predicted from a sequence of amino acids. Peripheral membrane protein is a protein that is found temporarily attached to the cell or mitochondrial membrane. Peripheral protein, or peripheral membrane proteins, are a group of biologically active molecules formed from amino acids which interact with the surface of the lipid bilayer of cell membranes. Unlike integral membrane proteins, peripheral proteins do not enter into the hydrophobic space within the cell membrane. Instead, peripheral proteins have specific sequences of amino acids which allow them to attract to the phosphate heads of the lipid molecules or to integral proteins. The ability to attach to the membrane but not be locked to it allows peripheral proteins to work on the surface of the cell membrane. Peripheral proteins can be activated or disabled through a number of different pathways. Many peripheral proteins are also a part of many complex biochemical pathways. They can be involved in moving substances within or outside of a cell, activate other proteins and enzymes, or be involved in cell-to-cell interactions. The main function of a glycolipid is to maintain the stability of the cell membrane. Glycolipids are also important for cell recognition and connecting cells to form tissues. So, what does a glycoprotein do in the cell membrane? Glycoproteins perform vital biochemical and structural functions. They enable cells to recognize another cell as familiar or foreign, which is called cell-cell recognition. They also help cells attach to and bind other cells, which is called cell adhesion. Carbohydrates attach to lipids, glycolipids, and to proteins, glycoproteins, extend from the outward-facing surface of the membrane. The principal components of a plasma membrane are lipids, phospholipids, and cholesterol. Proteins and carbohydrates attach to some of the lipids and some of the proteins. Carbohydrates on the cell membrane help cells function properly. Carbohydrates do this in 2 ways: helping cells recognize each other and providing protection for cells. Carbohydrates on the cell surface, known as the glycocalyx, are like identification badges that allow cells to recognize each other and differentiate between self and non-self. Most plasma membranes consist of approximately 50% lipid and 50% protein by weight, with the carbohydrate portions of glycolipids and glycoproteins constituting 5 to 10% of the membrane mass. Carbohydrate chains are attached to various proteins and lipids and modify their functions. The complex structures of carbohydrate chains, which have various biological functions, are involved not only in regulating protein conformation, transport, and stability, but also in cell-cell and cell matrix interactions.
So let's see what we learned. It is made up of a double layer of phospholipids that separates the cell from the outside world. The cell membrane's main mission is to serve as a barrier between the cell, which might also be a single-celled organism, and the world. So the cell needs to have a structure which allows it to interact with both. A cell's membrane is primarily made up of a double layer of phospholipids, fat-like, phosphorus-containing substances. Each layer is composed of phospholipid molecules that contain a hydrophilic, water-loving head and a hydrophobic, water-repellent tail. The heads in the outermost layer face and interact with the watery external environment, while the heads of those in the interior layer point inward and interact with the cell's watery cytoplasm. The region between the two layers is fluid repellent, which has the effect of separating the inside of the cell from the outside world. The cell membrane is semi-permeable, which allows selected molecules to pass into or out of the cell. It contains proteins that provide a number of critical functions. Since proper cell functioning depends on the movement of nutrients and useful materials into the cell and the removal of waste products from the cell membrane also contains proteins and other molecules that perform a wide variety of these duties. Some proteins are attached to these mats of phospholipids to help move nutrients, such as oxygen and water, and wastes, such as carbon dioxide. Some help the cell connect with and attach to the right kinds of materials, as well as other cells. And some proteins keep the cell from linking up with toxic materials, as well as the wrong kinds of cells, foreign or otherwise. Specialized proteins called enzymes help break down larger nutrients or help combine different nutrients with one another into more usable forms. Depending upon their design and function, protein molecules may be attached to the surface of one of the cell membrane's layers, or they may be fully embedded within the layer residing alongside the phospholipids. Some proteins tasked with funneling nutrients into and out of the space between the cell membrane's inner and outer layer cross only one of the phospholipid layers. Others, which are designed to transport nutrients into the cell itself or funnel wastes away from the cell, are large enough to span both. There are also proteins that help the cell maintain its shape. It contains carbohydrates that help to identify the cell and link the cell to others. Carbohydrates, compounds of carbon, hydrogen, and oxygen, such as sugars, starches, and celluloses, are found along the surface of the outermost layer of the cell membrane. Carbohydrates form glycolipids after linking with lipids, and glycoproteins after linking with proteins. Depending upon their design, glycolipid and glycoprotein molecules may act as chemical markers or receptors that help identify the cell or assist in linking the cell to other cells. Glycoproteins also bind with other proteins to make enzymes and other substances that, depending on the molecule's purpose, could be involved in blood clotting, capturing foreign bacteria, protecting against diseases, and other activities. Singer and Nicholson's fluid mosaic model is often used to describe the cell membrane structure. It can be difficult to envision how the cell membrane functions. After all, the cell membrane, and all the activities the cell engages in occur at levels too small for the naked eye to see. In 1972, two American scientists, S.J. Singer and G.L. Nicholson, developed the fluid mosaic model to describe the structure and functions of the cell membrane. The model notes that the membrane itself is fluid, in the sense that it is constantly changing. Individual phospholipids move about laterally in the same layer. However, one or more lipids may flip to the other layer on occasion. Lipids are drawn to one another through weak hydrophobic attractions, so while they do stick to one another, the bonds are routinely broken. The membrane's proteins also move about within this sea of lipids, as do cholesterols, which occur only in animal cells. Cholesterols increase the membrane's rigidity and firmness at moderate and higher temperatures by making the membrane less soluble. At lower temperatures, however, cholesterols separate phospholipids from one another so that the membrane does not become too rigid. The fluid mosaic model also describes how nutrients are transported into and out of the cell. Nutrient and waste transport may be passive, that is, it does not require energy, or active, that is, energy is required, to move molecules across the cell membrane. Passive transport can occur through diffusion, where molecules flow from a region of high concentration to a region of low concentration, down a concentration gradient. If molecules diffuse through a semi-permeable membrane, the process is called osmosis. However, in cells, a type of assisted passive transport, called facilitated diffusion, works because of transport proteins, which create membrane-spanning portals for specific kinds of molecules and ions, or attach to a specific molecule on one side of the membrane, carry it to the other side, and release it. In contrast, active transport is fueled by a coenzyme called adenosine triphosphate, ATP, which delivers chemical energy captured from the breakdown of food to other parts of the cell to move molecules up a concentration gradient. Among other things, active transport allows the cell to expel waste ions, such as sodium, Na+, from the cell, even though the concentration of sodium ions outside the cell may be higher than the concentration inside. And that's all for now. We've gone through the cell membrane. Next is the cytoplasm. And until then, don't forget to subscribe to our channel, and thank you for watching.